The overall goal of this study is to establish a mammalian cell-based in vitro Zika virus infectious culture system to study Zika virus replication, as well as utilization of the in vitro platform for drug development.
This step-by-step detailed protocol of Zika virus infectious cell culture system can help address the mechanism of Zika virus-mediated disease pathogenesis and evolution of viral virulence.
The main advantage of this technique is that it can allow for large-scale production of Zika virus for drug and vaccine development.
Begin by harvesting Viro cells at 80% cell density in a T75 culture flask by aspirating the medium from the flask and rinsing the cells with 2 milliliters of phosphate-buffered saline. Add 2 milliliters of 0.25% trypsin and incubate the flask at 37 degrees Celsius for 5 minutes. Following the incubation, add 8 milliliters of growth medium containing serum to inactivate the trypsin. Then, transfer the cells to a 15 milliliter tube. Next, remove 10 microliters of cells and mix with an equal amount of 0.4% trypan blue. Load the prepared mix to the cell counting chamber slide and obtain a count of the viable cells using an automated cell counter. Seed a total of 7 million cells into a T160 flask in a 30 milliliter volume. The next day, prepare an appropriate multiplicity of infection of Zika virus inoculum in 10 milliliters of serum-free culture medium per flask.
Safe handling of the virus is critical to prevent the spread of infection.